My sincere apologies. Okay, we'll just keep going. The respiratory microbiome we were going to talk about. We know now a little bit more about the respiratory microbiome, and although the respiratory tract is considered a continuum, we know that the microbes along the trajectory from nose to lungs slightly differ. We do know, however, that there are signals that are related to both health as well as to infections and inflammation. And crudely, you see differences in both diversity. So a more diverse microbiome is related to more health, a lower diversity to inflammation and to disease, and also the presence of potential pathogens and their overgrowth and outgrowth, as well as the predominance of oral type of anaerobic bacteria seem to be related with a more dysbiotic state. Let's go back to what we mean with that equilibrium or dysbiotic state. Well, from the ecological perspective, we know that if the microbes in a niche, which also counts for any human niche we're talking about, this ecological niche is at equilibrium. And from there, you have commensals that colonize it, and those commensals, as well as potential pathogens, can spread from their original ecological niche amongst the population. So it's an evolutionary trait that needs to be present to be able to share microbes amongst one another. So if this microbial ecosystem is at equilibrium, or we also call that eubiosis, it can resist or contain potential pathogens and thereby prevent them from causing any harm. The other thing it does is it's involved in immune modulation and in resilience. And resilience means if something happens to the ecosystem, it can quickly bounce back to its equilibrium or eubiotic state. However, we call that dysbiosis is when you acquire a new viral or potential bacterial pathogen, and you're not able to contain that, but instead this bacteria or virus can overgrow, invade, and or disseminate and cause inflammation. We think this is at the basis of lower respiratory tract infections. So how does that work? Well, there's three levels that your microbial ecosystem or your healthy microbes or commensals work. So this first level is that commensal microbes, in analogy to what you probably know from the gut microbiome, can compete with pathogens for the environment and for nutritional components. The other thing it can do is it can produce bactericidal and metabolic byproducts that can inhibit pathogen growth. Think about creating an acidic environment, think about production of short-chain fatty acids, etc. The second level of effect these commensal microbes may have is interacting with your mucosal or epithelial barrier between the outer environment and your body. We know these commensal microbes interact with this mucosa and can actually stimulate them to produce antimicrobial peptides, as well as help them in development of secretory IgA. A third level of interaction between commensals and the body, which helps to prevent invasion and dissemination of pathogens, is by interacting or communicating with our innate and adaptive immune system. For example, it helps us to get that balance between tolerance and adaptive immunity to pathogens. There's a fourth level that we will not go into depth into today, and that is that also the microbial community of our gut, which is the biggest reservoir of microbes and microbial function, is involved in protection of our respiratory tract through microbe-mediated and immune-mediated responses. But for today, we will focus on the respiratory tract. Okay, so let's go back to this, what we say, this state of our ecosystem that might either help to prevent infections or actually fails to do so. So to try to get an idea how that exactly worked, we designed a case-controlled study that was about 6-7 years ago, where we said, well, we want to see whether the microbial ecosystem of children with lower respiratory tract infections is really different from that of cases. As you all know, we use the upper respiratory tract to identify potential pathogens that might be related to infections. Think about the current swabs that we take in the nose and in the throat for detection of SARS-CoV-2. But we do that as well for RSV and influenza and the other viruses and bacteria. So we said, let's look in a very well-controlled case control study, a 1-to-2 design where we match the cases with 2 controls each in an age, gender, and time matching, which means that we could very carefully correct for seasonal variation in bacterial and viral pathogens shared in the community. So we established that study, but already quickly we identified that a part of our children actually quickly was eligible for exclusion because they had to move on to the pediatric intensive care ward. And we actually, in agreement with the ethical committee, we kept those children in a separate section of this study, which was called the Merrill study. And we were talking about 30 cases that had to quickly move on to pediatric intensive care ward because we acknowledged that those children were very important for us to study a certain aspect, namely to see or to prove that the microbial ecosystem in the upper respiratory tract is actually a very good proxy for what's happening in the lungs. So whether the theory I just presented to you seemed to be true. So for these 29 children, we asked the pediatric intensive care team upon intubation whether they would take materials from the lower respiratory tract, so endotracheal secretions, together with a nasopharyngeal sample. And here you can see the age distribution, and it comes very close to the neonates you are used to in your community. As you know, these children that require very rapid intubation are on average very young, so they were on average 2.2 months old with a quite small interquartile range. So as said, we looked at their microbial community in the upper respiratory tract as well as in their lower respiratory tract. And this is the average profile for the entire group. You see immediately in the color code that their microbial community on average is very similar. The microbial community in the upper respiratory tract in the nasopharynx is a little bit more diverse. And you see here with these purple arrows, several microbial types that seem to not hitchhike to the lower respiratory tract infection. And actually, there are the gram-positive commensals that we already had a keen eye on from the Corynebacterium, as well as the Dilysogranum group. These are commensals that are short-chain fatty acid and lactic acid producing bacteria and an analogy to the gut. Think about bifidobacteria and lactobacillus that are already very low abundant in these children, but are not hitchhiking to the lower respiratory tract. And also staphylococci in this group, we did not see back in the lower respiratory tract. But this is the average profile. So what we could do, because we had paired samples, we could say, well, in individuals, does this microbiome in the nasopharynx predict what's in the lung quite accurately? And we did that for pairs versus for random within that group of children. And you see that there was indeed a high level of what we call similarity or concordance for the microbial communities on person or individual level versus a very low of concordance for on group level. And to give you an idea, biological replicates have on average a similarity on entire community level of 0.71. So we approximated near equal communities in the nasopharynx versus the paired lung samples. And that tiny bit of difference was basically explained by absence of these bacteria. So basically proving our point that the nasopharynx is a good proxy in these very young children for lung microbiota at times of acute lower respiratory tract infection. So now we get back to the primary outcome of this study, which is the comparison of upper respiratory tract microbiota between cases and controls. So here you see we had a little bit more than 150 cases and twice as many controls. We missed only one control, and all these controls were sampled within 2 weeks following their identical case. So the paired case, which shows that the seasonal matching was very, very accurate. So you see already that in their characteristics, there are some differences between cases and controls, and it matches what's out there in the literature. Cases already had significantly more previous infections, previous wheezing, previous otitis episodes, and even previous hospitalizations. Also, cases had more often a very recent history of antibiotic use. They had lower levels of breastfeeding up to or beyond 3 months. And also educationally, seen the educational level of the parents was slightly lower, as well as their exposure to smoke in the environment was higher. So now move on to the viruses, because we know viruses are usually the trigger of lower respiratory tract infections in our young children. So if you look at any virus, we see that basically approximately 100% of all cases, we detected one or more viruses by quantitative PCR. But also in more than 80% of all our controls, we detected one or more viruses. So if you try to separate that out, you see that actually there was a significantly lower number of human rhinoviruses detected in our cases versus our controls. RSV, no surprise to this community, was significantly more often and almost exclusively identified in cases, as was a human metapneumovirus. The 2 years that were recruited, there were very low influenza seasons. So we found very little influenza, so could not see a significant difference here. But what we also saw that all these other viruses, the coronaviruses, adenoviruses, Boagart and parainfluenzas were not significantly more detected in cases versus controls. So let's now move on to the microbial ecosystem. And I show you this slide, which might be slightly complicated, but what we've done is that if you have one microbial ecosystem from a single child, we represent that by a single dot into this two-dimensional space to visualize possible differences between the two groups. So if two dots are very close to one another, they have a community composition that is very similar. If they are furthest away into this two-dimensional plot, they can't be more different. And on top of this, you see the biomarkers that seem to drive these communities. So communities that are driven by these gram-positive commensals that I was talking about, they are more on this side of this two-dimensional spectrum. Whereas if they're more driven by Staphylococci, for example, they're more on this side. Streptococcus pneumoniae and Haemophilus-driven communities are more on that side of the two-dimensional space. So I color-coded these cases in red and the controls in blue. And these ellipses are depicting a sort of the standard deviation around the median community composition. And what you see here is that these two standard deviations significantly differ, with a very significant p-value and an r-square of 3%, which means that the overall community compositions on average differ 3% from one another. That sounds maybe not a lot, but actually on a total community, that is quite big. So if you then try to classify, try to help us clinicians to see whether the viruses predict whether you're ill or not, we already knew that was actually quite poor in classification power. The area under the curve is only 70%. Well, if you use a total microbial community composition, that is actually slightly better, 0.76 or 76%, but it's still not perfect, isn't it? And as you can see that because in this previous slide, you saw that there was quite a lot of overlap between cases and controls. If you look, if you take the host characteristics, as I showed you at the beginning, actually that does even better, but we can't diagnose an infection based on patient characteristics, isn't it? So we were a bit puzzled at this stage what to do next. But then we had this brilliant student, Wing Homan, who said, well, actually, I think this is an excellent chance to use artificial intelligence and use a machine learning algorithm to see whether that machine learning algorithm can identify picking parameters from these three resources and see whether they can help us to identify who's actually sick and who is not. So that's what he did. And this is the overarching outcome. What he could do with this model with a 92% accuracy is identify who's a case and who's a control. And maybe it's not a surprise that having RSV is the most important marker there, but followed by maybe less, each individually less important, but altogether adding up to this 92% classification power, a whole bunch of microbes and host characteristics. So the microbes are here in green and the host characteristics are in blue. And what we observed was that the presence and abundance of non-typable Haemophilus was a good second marker, Streptococcus pneumoniae a good third, but also the absence of specific Moraxella species were part of this prediction model. And then also the absence of breastfeeding, the use of bronchodilators, and the use of antibiotics in the last 6 months. So that's how this classification model was built. And here is the second viral biomarker, which is basically the absence of human rhinovirus, which helped us to classify our cases. So it's a little bit of a paradigm shift, isn't it? But bear in mind, this was the overarching lower respiratory tract infection group. But as we know, there's different phenotypes in these groups. So that's what we try to identify as well. We asked an independent panel of pediatricians to separate out for us the pneumonia cases, the bronchiolitis cases, what we call a mixed infection. So they might have a bronchiolitis, but there seem to be also evidence for a bacterial superinfection, or the children that we call either viral-induced wheeze or wheezing illness. And we run again a prediction model. And interestingly enough, for all these 4 groups, we were able to, with slight differences between those predictive models, find a model which was highly accurate from 0.90 to 0.94, so 94% prediction of powder. And in 3 out of these groups, RSV was again a first, but not that outstanding anymore compared to the overarching group. Streptococcus pneumoniae was again a second in pneumonia, non-typable Haemophilus and Streptococcus pneumoniae were part of this bronchiolitis predictive model, and non-typable Haemophilus and actually a commensal Pseudomonas, part of the mixed infection model, but also again, absence of Moraxellas in pneumonia, absence of Moraxellas in Corynebacteria in the wheezing illness, and apologies, absence of these 2 gram-positive commensal categories of bacteria in the mixed infection group. So that is a slight paradigm shift. So we said, well, this is a bit difficult because generally we treat these children with antibiotics and these children not, and possibly we do treat these children with antibiotics, whereas these kids we're not treating with antibiotics. So how can we work with these data? So to get a bit of a better idea, we also said, let's look at this group of children that was hospitalized to see whether we found also whether these microbes predicted duration of hospitalization. So we build a model trying to predict number of days of hospitalization in these children. And of course, not a surprise, age was a first important predictor. The younger you were, the more likely you were to stay in hospital for a longer time. But then we found different microbes, and actually only microbes, not viruses, at the top, which help to predict duration of hospitalization. Again, a commensal Neisseria, which is an oral type of bacteria in your upper respiratory tract, whether there was Staphylococcus or not, et cetera, et cetera. And only the presence of RSV and the absence of human rhinovirus were at the very bottom of this predictive model. Well, how well did this model behave? Well, this is not a perfect one. So it's just a starter. There was a significant correlation, but you see there is quite some variance around this line between hospital duration and predictive hospital duration based on this model. However, we thought, well, part of these children were actually treated with antibiotics at day 0. So perhaps we should stratify the 2 groups, having received antibiotics or not. And the interesting thing was that if children received antibiotics, this correlation was totally lost. So the microbes at the start of disease did not relate to duration anymore as soon as you started antibiotics. But if you did not, the correlation became actually even better, which gave us the hint that indeed the microbes at time of hospitalization are drivers of inflammation and recovery. So another way to look at recovery is at ecological recovery. From all these children, we also obtained materials of their nasopharynx after 4 to 8 weeks. And here again, you see the microbial community, the average standard deviation in red at admission. And the purple ones, a little bit difficult to decipher from the blue ones, which is the controls, is where they are at 4 to 8 weeks after recovery. And although you see that moves to near the control group, it's not completely there. The p-value is 0.08. So there's borderline still a significant difference between their communities at time of recovery and that of controls. So we were quite interested in that. So we said, well, perhaps the fact that these children, or some of these children, have a slow ecological recovery, does that relate to their risk of having recurrence of infections? So we looked again at their time of hospitalization, their microbial communities. But this time we stratified of children that later on we learned to know had no recurrence, or that did actually have a recurrence of infection in the 4 to 8 weeks following. And bear in mind, these are mostly mild recurrences, although still about 30% got a new antibiotic course for that recurrent infection. And indeed, we saw that at time of hospitalization, we basically already saw that the microbiota of the children that were moving on to have a recurrent infection were different from that of the controls. And what we observed was, again, that the abundance of bacteria like non-typable Haemophilus, but also oral type of bacteria, including commensal fusobacteria and actinomyces, were higher in the children who ultimately had a recurrence in the 4 to 8 weeks following, whereas the absence, again, of these commensal gram-positive bacteria were also associated with recurrence of symptoms. So I think we start to see a pattern here, isn't it? So that to give you an idea what's happening at time of infection. So let's now move back, because we've always had an interest to see what the microbiome had a relation with development of infections, because if that's the case, we can see in the long run whether we can find preventive strategies to prevent children from getting these infections. So that brings me back to the first 1,000 days of life. As you all know, the first 1,000 days of life is very important for children. Basically, not only their body, but also every organ system in their body is undergoing very rapid growth and development. But thinking about that, all these organ systems at time of delivery are basically already sufficiently developed enough to function. Not so our microbiome. We do regard nowadays our microbial ecosystem, or the human microbiome, as one of our organ systems, very important for, as said, barrier functions, prevention of infections, and immune maturation. However, when a child is born, it only needs to start developing this additional organ system. It's not yet there. It only starts to accumulate from that point on. So you can imagine that needs to happen quite rapidly. But it depends a lot more on the environment, on how you're born, what microbes you get from your mother. A vaginal delivery versus a C-section is very different. How you're fed, do you receive breast milk, or do you receive formula milk? Do your siblings cuddle you and share their bugs with you at the time or shortly after delivery? Do you receive immunizations? Do you get an early life infection? Do you receive antibiotics? Season, it all will matter what you're exposed to as a child. So also what you can pick and choose from to develop your own microbial community. And the idea is that if you are able to very rapidly develop a balanced microbial community that's more stable, it might also be more infection resistant and resilient, which will not be the case if your microbial community has a bit less opportunities to develop to a diverse and stable community. So to try to understand that, in 2014, we started a prospective microbiome study, which was called the Microbiome Utrecht Infant Study, or MOUSE, and MOUSE is the Dutch acronym for a mouse, a little mouse. And we recruited approximately 120 children during pregnancy, because that allowed us to take the first biosamples directly after birth, which managed for most of the children. We had biomaterials from the skin, from the nasopharynx, from the oral cavity, and the gut within 2 hours after delivery. And then we obtained materials at day 1, week 1, week 2, month 1, 2, and then we took a bit bigger intervals, because we anticipated that microbial community would have the biggest changes at those early beginnings, and then might take a little bit slower pace along. So for now, I'll focus the first data on the nasopharyngeal community development in those children, because we had seen in the previous study that that seems to be related to acute infections. So here again, you see this two-dimensional plot where each dot is one sample from a single child across 12 months of life. And we color-coded them according to day, week, month that we obtained and whether we obtained samples during acute respiratory infections. Again, each dot is representing one community composition. If they're very close to one another, they're very similar. If they're far away, they're quite different. So again, the earliest time point started over here with quite a big spread. You see this wide confidence interval, this rapid sort of caterpillar-like movement across time, which gave us the first idea that, wow, all these children seem to develop according to a specific pattern across time. But the widespread, the bandwidth of this standard deviation was quite wide, and definitely here you could arrive here halfway this caterpillar either after 2 weeks or 4 weeks, but also as late as 6 months. So we got this idea that possibly this pace of development might be a clue to stability as well as to resistance against infections. So first we looked at what is actually related to the pace of this development. Well, and besides age, that was having siblings in a household, which is interesting because we know they share their microbes, going to daycare from early in life on. And in the Netherlands, children go often to daycare as early as 4 months. Season of birth, whether you receive breast milk for 3 or more months, mode of delivery, and antibiotic use in the last month. But when we look at that spread at those very early time points, we even see that the weight of these individual factors change over time. For example, season is very significantly related to that early seeding at day 0 and quickly wanes over time, whereas, for example, birth mode and antibiotic treatment seem to be quite significant across that time. And, for example, breastfeeding actually only starts to kick in after week 1. So you see that there is even the influence of these factors will vary across time. So we said, okay, let's see whether the pace of this development trajectory is related to susceptibility to infection. So we looked at our cohort. I looked at the normal distribution of the number of infections these children had. Bear in mind, they were all term-born babies without complications. So number of mild to moderate infections was what we could use as a readout. And here you see we had this nice normal distribution from 0 to 7 infections over the first year of life. So we said, okay, let's use this distribution to stratify our cohort in average, below average, and above average number of infections. And then let's look back at the microbial community development. So for this graph, we actually were able again to calculate a microbiome age with an artificial intelligence model versus chronological age. And actually the model did very well. We based that on this green group, which we call the reference group, and we observed that we could predict or build their microbiome age quite closely to their chronological age in the green group. It's nearly a straight line. So if we then compare the yellow group with that green group, we see that for the first, let's say, 60 days of life, they develop, their microbiome age develops in a similar page. But as soon as the first infections depicted here in orange start to kick in, you see that they accelerate a little bit until the sort of third trimester of the first year of life, they start to come together again. But the most interesting pattern we found in this orange group, the children who had recurrent infections in the first year of life. Again, those infections were all following mostly 2 months of life. But what we observed was that, especially here in those first 60 days of life, we observed that the microbiome age accelerated very fast compared to these other 2 groups. So if we zoomed into what was happening there in those first 60 days of life, and compared again these 3 groups, what we observed was that especially, again, these gram-positive commensal bacteria I was telling you about, analogy to Bifidobacterium and Lactobacillus species in the gut, but these are very respiratory microbiome specific, were very early and throughout the first year of life abundant in children with very little infections, whereas they popped up a bit later, or actually not at all, in these other 2 groups, and they didn't persist as well. And during infections, you actually see that they're hardly present there, which, if you remember, is in analogy to the data I showed you from these children that were admitted to the pediatric intensive care unit with an acute respiratory infection. And if you then correlate the presence and abundance of these gram-positive commensals with potential pathogens from the pneumococcus family and the non-typable Haemophilus family, as well as these other signatures that previously we observed to be associated with an acute infection, so oral flora, including Neisseria species, we see that there is a negative correlation. So they were increasing with these two groups, and they had a negative correlation with presence and abundance of these two groups of bacteria. So, as said, the nasopharynx seems to be a very good proxy for the communities that represent also the lower respiratory tract in very young children. But there were some signals from the international community that said in adults, actually, the oral communities seem to play an important role as well. So we said, well, let's have a look at that, because we also obtained saliva samples, which in children that have no teeth yet is a very good proxy for also what happens in the oropharyngeal community. And here you see, this is only for the first 6 months, the microbial community in the oral cavity versus those in the nasopharynx. And the color code already shows you this is very different. There's a predominance of oral type of strep over here in the oral cavity, whereas if you remember, at the beginning, it is Staphylococci, it's Corynebacteria dalysigranulum, and later on also Moraxella that's popping in the nasopharynx. So again, this student, Bing Ho Man, who's now halfway his residency for pediatrics, said, let's have a look whether I can get a better idea about how these two communities correlate with one another. So first he looked at stability of the microbial communities in these two niches over the first half year of life. And what you see is that this is a reversed measure. You see that the community in the nasopharynx is at the beginning not very stable, but then reaches stability quite quickly. And then you see it sort of hangs around this 0.5 measure for the rest of the first 6 months of life. What you see in the oral cavity is that from the start, the microbial community is much more stable and also remains more stable over that first half year of life. So as a consequence, we could actually not directly link the microbial community of the oral cavity with risk of infections or a number of infections over time, whereas as I showed you, we could do that for the nasopharynx. But we know that at times of infections, there's an emergence of oral microbes in the nasopharyngeal community. So he said there must be a link between the two. So he said, what if, this is a microbial ecosystem landscape. This is a theory that has been postulated in ecology, that if you have very stable ecological systems, different niches have their own equilibrium and they have very much difficulty to move into one another. However, at times of dysbiosis also counts for a forest, for example, you see that temporarily these ecosystems might have this tendency to move into one another, which they call loss of topography, loss of ecological topography. So he said, let's see whether I can look at that potential loss of topography between the nasopharynx and the oropharynx in our children as well. So to do that, he looked at the distance between the nasopharyngeal community and the oropharyngeal community in these matched samples from our children from day 0 all the way to 6 months of life. And at the beginning, and he stratified that between children that had very little to no infection across the first year of life and the ones who had more than 2. And what he observed was that at the beginning, first 50 days of life, that the distance between communities was similar for children with or without risk of infections over the first year of life. However, around this time, we observed that the children with a lower number of infections had actually more distance, more distinct communities compared to the children that ultimately had more infections. And at times of infections, that's that dotted orange line, we saw specifically after 150 days, at times of infection, we saw that there was a near crash of a topography, and these two communities started to really trickle into one another, which might explain why we start to see popping up oral commensals in the nasopharynx at times of infection. And to give that a better idea, what we saw is if you look at very oral specific, what we call oral indicator species that during health are not present in the nasopharynx, they indeed pop up during infections in the nasopharynx, and nasopharyngeal indicator species actually do not pop up in the oral cavity. But the interesting signal we saw here was that actually on average a month before a symptomatic infection, these oral species already popped up in the ecosystem of the nasopharynx without any symptoms. So they preceded the factual infection. So that was an interesting clue for us to have a better idea about what to do next. So in summary, again, what we observed was that around birth, communities are seated very similar in the nasopharynx and the oropharynx. They both become a niche-specific communities, so they become distinct, they become distant from one another, and that's sort of at a maximum around 3 months of life. However, in the children that develop further infections in the period following, we see a less distinct niche differentiation, and at times of infection, their niche distinction collapses. And this is associated with low, again, gram-positive commensals and more predominance of these oral bacteria in the nasopharynx. Okay, so now let's move on. See if I have a little bit of time. I think I'm fine. What we did, as said, we obtained upper respiratory tract material from our children. But what we also did, and besides looking at the microbiota of these children, we also were able, in a subset of these children, to look at mucosal gene expression. And in all these children, we also determined their viral data. Again, not only during infections, but actually also during absence of symptoms. And because these are paired, we could look on group level whether microbiota are related to gene expression and to viruses, but also on individual level. And what you then see is that on individual level, you see a very good matching between gene expression and microbial communities. So these two seem to be really closely linked with a very high correlation of 11% explanation of one set by the other. On group level, we saw that as well, but not as distinct, which means that really on individual level, the microbes present seem to be linked with gene expression profiles of the mucosa. And interestingly enough, the viruses were not so much related with microbiota at the time of presence, and also only modestly with gene expression. Okay, so let's try to look at these three distinct sets: microbiota, host gene expression, and viruses. So getting back here at the microbial community development, which we linked to number of infections over time, what we did is we tried to dive a little bit more into nasal profiles, or fingerprints, as sometimes people are calling them. What we observed was that if you look at clusters, profiles, fingerprints, you see that children move across those fingerprints over time. There's this pattern that children move from either a staph or strep-dominated communities to other staph communities. Again, these Corynodolo, Moraxellas, Haemophilus, Neisseриas, etc. But keep in mind that we will work now with these profiles or fingerprints. If we correlate again those fingerprints to number of infections, of course, they need to be again related, like I showed you in the previous data slides. Again, Corynodolo profiles are highly linked with the green group, absence of infections, whereas the other ones are less so, and this is significant. Movement from a Corynodolo to a Moraxella profile, especially in early life, was significantly related to a high number of infections in the consecutive year of life. And again, Haemophilus abundance, so moving to a Haemophilus-dominated community, was related to 3, 4, and 5, and 7 infection groups, but was rarely seen in the children that had only none to very few infections across the first year of life. And again, the biomarkers of these profiles were again related to number of infections, with Dodicigranum and Corynebacteria associated with no infections, and a very early predominance of Moraxella. And across the first year of life, Haemophilus and Streptococcus, which is we identified as Streptococcus pneumoniae, are related with more infections. Okay, keep that in mind. We're moving forward to the viruses. So again, like we've done with the case control study, we detected presence of viruses during all these asymptomatic episodes, as well as at times of infection. And what we observed was that already quite early in life, many children were carrying a virus or even more viruses. And we saw in the first week of life, already human rhinovirus popping up. Again, asymptomatically, but we did detect them. And bear in mind, the first symptomatic infections really started to kick in from at the end of month 2, beginning of month 3. So human rhinovirus a lot, but also these other viruses started to pop later. So we said, well, especially these asymptomatic viral detections, are they related to your susceptibility to infections later on? And we did indeed see that time to first viral acquisition was significantly higher in the blue group, and I apologize for the small print, which is the 5 to 7 infection group compared to the other 2. And again, the 0 to 2 infection group had the longest time to first viral infection from all 3 groups. Well, if you go again to a little bit more mathematical pattern, you see that the first viral detection was done on average around 60 days, and it ranged from 0 to 180 days. The first RTI symptoms were starting on average only around 130 days. And the end of the first symptomatic episode was actually, of course, a bit later. So if we looked in the pair data between whether first viral detection was always or mostly preceding symptomatic infections, that was the case. It ranged from 0, so viral detection was either coinciding with the first symptoms, to 260 days, so preceding first symptoms with 260 days. But look, there's not a negative number. So it was never the other way around, that viruses were detected after the first infection. So there was this bandwidth between detection of the first virus and your first symptomatic episodes. Again, park that a little bit, but I'll come back to that in a couple of minutes. So now the mucosal gene expression profile. We did basically the same thing as we did for the microbiota. So looking at the mucosal gene expression pattern as a single dot in a 2-dimensional space. So again, each dot here is the mucosal gene expression of a single sample, okay? And here you see that you can cluster these. You have multiple clusters, some big ones, some smaller ones. But if you then look at the time of sampling, you see that certainly at day 0, day 1, week 1, you see that these clusters are going hand in hand with the time of sampling. However, slightly later, you see it all starts to trickle through one another. You can be in this cluster either at week 2, but also only at month 4. So we saw this sort of development pattern in mucosal gene expression, but after week 2, it was again, some followed a fast pace in mucosal gene expression and others not. So looking at that pace of mucosal gene expression, we did a separate analysis and looked whether we could find modules of genes that were co-expressed across time. And we found 4 big modules. Module 1, which was only temporarily increased in expression at day 1, but then again inhibited until month 6, and then it was vastly overexpressed. Module 3 did something similar. We saw that it was suppressed in the first 2 months of life, and then it started to become overexpressed over time. And module 2 and 4 did something opposite. Module 2 had a high expression at the beginning of life and a low expression at the end. And module 4 had a high expression from week 2 to month 4 and then down again. And if you look what on average these modules express, so module 1 is a module that contains all TLR cascade genes and neutrophil activation pathways. You see them again in order of importance. Toll-like receptors, interleukin 4 and 13, and neutrophil degranulation, etc. Whereas module 3 was actually exclusively interferon pathway genes. So you see expressed late, but not so much early, a little blip here. So probably the interaction with the first microbes gave a little bit of a rise in expression, and then it was dampened again. whereas module 2 and 4 were actually containing epithelial homeostasis and maturation and differentiation pathways, which were expressed at the beginning of life and downregulated later. So that's interesting, inflammation-driven and more homeostasis-driven modules that have a counter regulation across time. So then we said, let's look whether this transcriptome across time seemed to be linked with the microbes present at that time. And we performed a HALA analysis, which can take the biomarkers from the microbes related to your mucosal transcriptome. And what we observed was this set of microbes standing out. This is not determined. So this was objectively acquired, and it picked, again, Moraxella, Haemophilus, Streptococcus pneumoniae, and an oral strep on this side, which were associated with upregulation of this group of genes and downregulation of this group of genes, whereas, again, Corynebacterium and Dolizigranum followed the exact opposite pattern. They were related with downregulation of this set of genes and upregulation of that set. And if you try to zoom in again, what you see is, again, with a different approach, that genes associated with high amounts of Haemophilus and strep across the first year of life, downregulated with these Coryna and Dolizigranum bacteria, are again these neutrophil degranulation genes, leukocyte migration genes, and all these inflammasome-related genes. Negatively associated again with Corynebacteria were also interferon, a group of interferon-related genes, whereas related with oral type of strep were cilium and microtubules, so homeostasis-related genes, and related with Moraxella in a later stage, so from 2 months on, were more adaptive immune-related genes. So you start to see a pattern of microbial communities versus host transcriptome. So that model 3, which was interferon driven, we were quite triggered by that, because we said, well, is that in some way related to first acquisition? Even we know that first acquisition of viruses is asymptomatic. Are they related? And indeed, we observed, if you have first viral acquisition under 1 month, this upregulation of this module 3 was speeding up rapidly in this first 60 days of life. If your first viral acquisition came a bit later, it was a little bit slower. And if it was only following month 3 or you had no virus, you see that that actually only developed later, and it took a lot longer time before it was that same level. So again, checking whether these modules were then associated with ultimately symptomatic infections, we saw again that module 1 was from about 120 days on related to symptomatic infections, module 3 as well, but that started earlier, whereas module 2, this homeostasis module, was related with absence of infections. And here, if you associate none versus 5 to 7 infections, it's even more clear that children with ultimately recurrent infections had upregulation of module 1 already from about 50 days of life, and actually module 3 was upregulated from day 0 on. Trying to tease out all these little dots, the correlation between these biomarkers of microbial biomarkers with whether there was a virus or no virus, we see again that early Moraxella was associated with early viral presence, Haemophilus again as well, Streptococcus pneumoniae also associated with an early presence of viruses, whereas oral strep had only a tiny window of association with no virus. And then the fourth one, again, the biomarkers, bacteria associated with infections, as you know, early Moraxella only associated with recurrence. And throughout the first year of life, Dolizigranum and Corynebacterium associated with no infection. And again, Haemophilus and Strep pneumoniae from very early on associated with infections. And why do I show you these little dots? Well, to try to tease out this triangle of interactions. So what we think we can conclude is that in this very early window of life or window of opportunity, if you have early and prolonged gram-positive commensals, which go hand in hand with absence of early life viruses, it goes hand in hand with an upregulation of homeostatic mucosal gene expression, Whereas if you have early viral acquisition, this goes hand in hand with early Moraxella, early and prolonged Haemophilus and Streptococcus pneumoniae, eukaryotes, also the presence of these oral type of bacteria, so loss of this niche differentiation, as well as with more inflammasome and interferon triggering and reduction of homeostasis gene expression. And we know that this triangle seems to be related with a number of symptomatic infections across the first year of life. And of course, now we want to know whether that is also predicting their long-term health. So these children have now reached 5 to 6 years of life. So we're going to look at how these children developed and whether we can associate this with both atopic constitution as well as lung function. First data from Australia actually show similar connections between what we see in early life and these long-term atopic and wheezing phenotypes. So we think it's very useful to know this information. And if you want to know whether there's already intervention trials tried, very little. This is a group that just reported this paper where they used in a mouse model both Dolizigran and Picarum, and in a previous paper, Corynebacterium pseudodiphtheriticum, and challenged them with both either an RSV or a pneumococcal challenge. And indeed, they observed that if you had these Dolizigranum in the previous paper, Corynebacterium added to your respiratory tract, they had lower RSV titers and lower pneumococcal counts, as well as lower amounts of pro-inflammatory signals. So I think it's a first signal to show you that the field is moving forward, and hopefully we can start to get an idea about how early life microbiome is associated with respiratory health, how we can use that both from a diagnostic perspective, also from a treatment perspective, as well as hopefully from a preventive aspect. And of course, there would come into the equation that we need to start to find ways to promote healthy microbes instead of trying to eliminate harmful bacteria and viruses only. Leaves me to thank my team, and in red, Erling Houman and Wouter de Steenhuizen-Pieters and Emma de Koff, who actually were responsible for the data I showed you today, and my two colleagues from the Netherlands, Lieke Sanders and Marlies van Houten, who were co-PIs of these Dutch studies that I showed you, and of course the funders of previous and current work. So I hope I stayed within time, and I think that leaves questions, I hope now, and let me try to stop sharing. Thank you.
Debbie, thank you very much for a fascinating talk, and congratulations on an amazing body of work. We do have time for questions. Does anyone have any? Chris, I think you were first, the first one I saw anyway.
Thank you. That was a fantastic presentation, incredibly interesting. I just have a quick question about the first part of the work that you presented from the case control study. How do you see this being applied in practice? Is there a way to develop a simple test to predict which children are going to benefit from early antibiotic treatment, and which can safely go home from A&E or from the general practitioner? And just any comments you have on that?
Could I just for the people online, Chris asked, how is this going to be, how are we going to use this information in practice? How are we going to be able to identify individuals at risk?
Yes, I think it's an excellent question, and it's exactly what we have in mind, although you have to be careful, of course, with this type of work to immediately jump to conclusions. It seems promising that you can you have a much better predictive capacity if you compare it to, for example, the standard diagnostics we use at this moment. And we do feel we need to have a more balance instead of using phenotype-driven decisions, whether you will give or provide antibiotics. For example, if you have bronchiolitis, you try to not do it. And with pneumonia, you should always do it. We think that your microbial community composition might help you to take that decision, weighed, of course, with clinical aspects, and also it might help you to know what's present there, so maybe what to target and not to try to tray as small as possible. We also think that aspect of trying to predict who does recover rapidly, spontaneously, might help in that equation. So I would imagine that if you can predict that a child will be struggling for 14 days, despite having what we call clear viral picture, that you might be earlier in trying to intervene compared to a child that has this predictive capacity of recovering quickly. But that said, we have only one study that we were able to do this modeling on. We're currently busy with actually replicating this in one of the PERCH cohort studies from South Africa. And of course, it's for us also very interesting to see whether we see similar patterns, whether these are universal or very site-specific, to see how we can move on with our advice over time. But yeah, we do hope it will add to a less crude decision making of who to treat and not, and with what type of antibiotics.
Thank you. Neil.
Yes, thanks very much indeed. One of the questions that comes up from my side is, what is the influence of chronic disease on this relationship that you're showing between commensals and pathogens? For example, if you have a child who has bronchopulmonary dysplasia, are they growing up with already a poorly defensive
Thanks, Neil. So just for those online, Neil's asking about the influence of chronic disease such as bronchopulmonary dysplasia in the development of the microbiome, the respiratory microbiome.
Yeah, it's again an excellent question. And I can say we are currently trying to investigate that in collaboration with Professor Boortman, also present in your location, in one of his cohorts. And of course, there will be interesting to tease out what comes first and what next. We also know that the microbial community will adapt following inflammation, following damage, following antibiotic treatment. So to tease out what comes first and what next is important, as well as what you're doing with that information. We have published a study as well on a birth cohort in children with cystic fibrosis. Born with cystic fibrosis, and what we observed that they have a different community composition compared to non-CF babies, although the community members are very similar. So the balance in their communities is different, and probably that's because of a different environment. What we did see is that they only started to deviate further away with reduction of healthy commensals and an increase and selection of gram-negative pathogens following their first antibiotic exposures. So again, timing of where you look and what the target and the goal is, why you want to look with your microbial communities needs to be well defined. And if you want to prevent dysbiosis, that's another question, and whether you want to use this information to treat your patients. But yes, you can use it. We have done it. Community members are definitely in early life not very different in chronic disease, but the balance within those communities seem to be slightly different depending on their mucosal environment.
Okay, we've got time for one more quick question. Nigel.
Thank you. I know you mentioned at the beginning a relationship between the gut microbiome and the respiratory tract microbiome. I just want to explore a particular risk factor for that, which is the use of antacid medications. And we know that those influence the gut microbiome. Have you explored a relationship between exposure to antacid medications and the respiratory tract microbiome or development of later respiratory tract infections?
Thank you. So just to recap, that's a question about the relationship between the gut microbiome and the respiratory microbiome and the influence that drugs and medications that we know can influence the gut microbiome, such as H2 blockers, which block the acid production in the stomach, how that might relate to the subsequent development of the respiratory microbiome.
Yes, I think it's a couple of questions in one, isn't it? So first of all, how the gut microbiome relates to the respiratory microbiome. Yeah, given time, I had to choose a little bit what I should pick, and we were focusing on the respiratory microbiome, but hopefully soon. We're currently having a paper in revision also of this cohort where we were able to construct a network across the body of microbes to understand whether microbes in different niches were linked with one another. And we did find very clear networks of microbes that seem to be coexisting in the different niches in specific children. And even that network and the strength of that network seem to be related to their susceptibility for infections. How they influence one another. So whether, for example, antibiotics would eliminate commensals in the respiratory cavity and not in the gut. I don't think we can model that based on children because they would all be similarly affected. How antacids do that, again, I think they will change the environment in multiple niches. So not only that of the gut, to tease out that effect. And if you want to know whether we have a study looking at the effect of antacids on both the respiratory community and the gut, we unfortunately have none. It would definitely be interesting to look at that. And I definitely think it will affect both the gut microbiome as well as the connection across the human body. And it makes it even more complicated because we also know that the gut microbiome on its own will have long distance effects on the respiratory immunity through regulatory T cells. So the teasing out who's exactly doing what and whether you can actually separate that out from one another is a very difficult question, I think. And I unfortunately have no answers for that at this moment, but it's definitely an interesting one. And one that we should pursue. For now, we have pursued mostly the effect of antibiotics because we saw, especially in very early life, a devastating and long-term effect on microbial ecosystems. So that's at the moment one of our other foci of research.
Debby, thank you. I know we could ask lots more questions, but unfortunately we've run out of time. On behalf of the Society, I really want to thank you very much indeed for your fascinating talk, and we really look forward to hearing more your work in the future. Debby, thank you.
Well, thank you as well.